A certain toxin-producing gut bacteria may be responsible for triggering both the onset of multiple sclerosis and ongoing disease activity, according to a new study led by a team of researchers at Weill Cornell Medicine and NewYork-Presbyterian. The team works with investigators from Cornell's Ithaca campus, as well as the California, San Diego, University of California, Davis and Pittsburgh, and has a long-standing collaboration with scientists at Rockefeller University. The study, published February 28 in the journal Clinical Investigation, identifies Clostridium perfringens, which produces an unusually high amount of Epsilon toxin in the gut microbiome people with MS. The study continues to show that in a preclinical model MS, Epsilon toxin opens blood vessels of brain, allowing inflammatory cells to access central nervous system and cause demyelinating characteristic MS. MS is a disabling disease of the central nervous system that usually begins in young adulthood and affects approximately 1 million people in the United States alone. Early in the disease course, MS is characterized by episodic recurrences and remissions of neurological symptoms such as vision loss, weakness, and instability. In the later stages of the disease, MS tends to progress in approximately 40% of affected individuals, despite numerous advances in treatment. D. Co-senior author, Professor of Neuroscience at the File Family Brain and Mind Research Institute at Weill Cornell Medicine. MS has many mysteries, said Timothy Vartanian. Why do some people get MS, despite similar or identical genetics? What explains the episodic nature of relapses and remissions? How is the central nervous system targeted, and why is myelin specifically targeted? Clostridium perfringens and Epsilon toxin may explain many of these mysteries. An environmental trigger is required for MS to occur in a genetically susceptible individual. An abundance of Clostridium perfringens, which produces the Epsilon toxin in people with MS, suggests this may be culprit. Epsilon toxin-producing strains Clostridium perfringens live small intestine, and Epsilon toxin is only produced briefly when the bacteria is in a growth phase, keeping with relapsing nature MS. Perhaps most strikingly, Epsilon toxin specifically targets cerebral blood vessels and myelin, providing a clear picture its mechanism action. Despite growing evidence that Epsilon toxin-producing strains of Clostridium perfringens may be relevant environmental pathogens for MS, modern studies of the gut microbiome in people with the disease have failed to detect these strains. In current study, Yinghua Ma, an assistant professor of neuroscience research in Vartanian lab, led work with co-authors David Sandino and Jennifer Linden from Brain and Mind Research Institute, showing that more sensitive techniques detect them easily. Strains in the MS gut microbiome. Previous research used to use a method where you could see types of bacteria that were there, but you couldn't really see toxin or more functionally relevant parts of species, said Professor Christopher Mason, Division of Physiology and Biophysics, and co-director of the World Quantitative Prediction Initiative at Weill Cornell Medicine. Using highly sensitive DNA detection techniques, Ma found that people with MS were more likely to carry C. perfringens, which produces Epsilon toxin, in their small intestines than healthy controls. The team brought in all-weapons modern molecular biology to address question pathogenesis and possible drivers multiple sclerosis, said Mason, who is also a professor in neuroscience at Weill Cornell Medicine Brain and Mind Research Institute. The researchers who established this correlation tested whether the toxin alone would cause the disease. For this, they turned to a standard mouse model of MS, in which animals are predisposed to autoimmunity, but MS-like disease only occurs when mice are given pertussis toxin. Ma replaced pertussis toxin with Epsilon toxin, and the animals developed a disease more similar to MS than previous models. The finding that Epsilon toxin can replace pertussis toxin in a mouse model MS is very exciting, said co-author Gregory F. Sarnenberg, MD Roberts Family Associate Professor of Medicine, and a member Gilles Roberts Research Institute in Inflammatory Bowel Disease at Weill Cornell Medicine. Not only does it develop a more appropriate model to study MS, but it critically identifies a novel microbial-derived marker that causes impaired immune privilege in the central nervous system to initiate demyelinating disease. Epsilon toxin functions earliest stage MS lesion formation, said Vartanian, who is also chief of the Multiple Sclerosis and Neuro-Immunology Division in Department Neurology at Weill Cornell Medicine, and a neurologist NewYork-Presbyterian Weill Cornell Medical Center. A treatment that neutralizes the epsilon toxin can stop our patients' new disease activity much more effectively than current treatments that suppress or modulate the immune system. In the short term, we act a sense of urgency to provide people with MS more effective and safer therapeutics, said Vartanian. Dr. Tim Vartanian and Dr. Yinghua Ma is co-founders and equity shareholders of Astoria Biologica. Dr. Vartanian and Dr. Ma are inventors of the patent application for method used to diagnose and treat multiple sclerosis.